I think we're going to get started, and I want to welcome you all. I appreciate your being here. I think this is going to be a fascinating kickoff to a project. It's going to be a lot of really valuable and useful stuff. And I've got a number of things that we're going to be talking about. Largely, we're going to be talking about a video project, but we're really going to be setting up you guys are a core people who are interested. There are lots of people who have let me know that they are interested, but they can't participate in this meeting. And so I want everybody here to feel free to ask lots of questions and make lots of comments, because especially the comments of, I don't really see how that's going to work, are really valuable. And they either bring on an additional explanation that clarifies to other people, or they show different directions that we really need to be working on. And I appreciate any input. I want to, David Freely isn't here, but I want to send kudos to him because he's been working on a lot of the online documentation that's really coming along, and there's a lot of interesting stuff, and I encourage you to go to the website and take a look at what's going on. For this meeting, I want somebody to volunteer officially or unofficially to be the producer, which is I'm going to be talking along, and I'm not in a good position to notice who's got their hand up to ask a question or something. So somebody I would like to keep in charge of making sure that when there's a question, we get to it and a comment. So who wants to take on that
I'll do it. Just raise your hand in the message. There's a message area that you can type something into.
Okay, that will be perfect. And then you can alert me when I need to be alerted.
Yep.
There we go. And two other things. I posted links in the beginning of the chat. There's a link to the PowerPoint that I'm going to be presenting, and there's a link to the
GitHub.
Yeah, to the GitHub, so that you can get the code if you want to load the code and follow along. And it's current to what I'm going to be showing. So I wish you all to participate at whatever level you feel like participating in.
And I actually don't see a PowerPoint.
It's the link is there. It's futureaisociety.org/MP files. No, it's the very first comment I put up.
We don't get because of the way Teams work, we won't get any past messages when we joined.
Oh, how unfortunate.
That's Teams. That's Teams.
Okay. Let me just put this. I'll put the PowerPoint up. The GitHub you can actually dig up pretty easily. Where would I find? There we go. There's the link to the PowerPoint. Okay, there we go. There's the PowerPoint. How about now?
I see it.
Okay. Okie dokie. Okay, now I need to remind myself how to share my screen. There we go. It's the share button, and I want to share that screen. There we go. Okay, we're doing an online conversation. That's us. Please raise your hand or make some sort of comment if you need to ask a question. But I'm going to give you the overview and agenda first. So save the questions until you see where they fit into the overview, into the agenda of things. We know that the neocortex structure must be based on some general rules, and we're going to be reverse engineering what those rules are as part of this project. And what we're going to be doing is, the intent here is to make some better AI. That's my intent. If you're interested in neuroscience, that's a great approach too, and I'm happy to participate in that conversation. But I'm not a neuroscientist, so I'm not in a be able to contribute to the neuroscience knowledge. And as far as I can dig up, I'm pursuing vision, not because I'm really excited about vision, because I'm excited about the whole thing. But vision is currently the best explained area as we go through. You'll be able to see that there are a lot of things that we know about how the brain works, and there are a lot of things about, although the specific connections aren't known, but many of the AI algorithms have limited generality. So if you are working in AI and you've got a vision system, that probably is not going to work for speech recognition. But I would anticipate that when we are working with real human intelligence, that speaking and understanding text and understanding hearing text are going to be more or less interchangeable, that the same algorithm works for lots of things. And that, I think, would be nice to find out. And so I'm pursuing what might be a general algorithm, and I'll talk about how this is going to work. And let's see if we go here. There we go. And you know what? I'm working on a PowerPoint and I haven't shared it. Is that correct?
I can see the PowerPoint and I can see the link.
Perfect. So it's on a different screen, so we've got a different place. Okay, we're all working perfectly. So I want to talk in terms of biological plausibility, and here we're talking about neurons and synapses and learning, and I will just want to wave my hands over that, and when we get to it, I'll spend about 5 minutes on it. We're going to be talking about compatibility with the Universal Knowledge Store, which is the software that we've been working on. And one of the things that I've already found is that I have to tweak here and there. And I'll show you one change that I had to make in order to make what I've already discovered compatible with the UKS. And that's part of the point of this, is that you try and implement something and you say, well, this isn't going to work, so we'll make some changes. I would like to make sure we understand that although many of you may be AI people, that we're not talking about any gradient descent backpropagation, not because we don't love gradient descent backpropagation, but because there is no possible way that it could work in the brain. And there is we can talk about, if you like, there is a backpropagation which is alleged to exist in the brain, but it turns out that there is it's a reuse of the word, and it's actually a different thing. The timing and the spike count is going to be extremely limited. When we're working through vision, you can you can recognize something in on the order of 1/10 of a second. And what that means is that the number of neurons that can be successively involved in the vision process is very, very small from a computational perspective, because neurons take at least 4 milliseconds to get off the ground and do something. And we don't have much time in the 1/10 of a second that you have. So we're going to be looking for ways that this can be extremely efficient. You have very limited visual resolution, it turns out, and I'll be talking about that when we get to it. And finally, I've got some initial development that I can show with some interesting conclusions and some interesting questions. So I've already had to modify the UKS code a little bit to make things compatible with what I can already observe as working. Okay, any questions before we jump in?
Do you ever think about the spikes as a form of combinational logic? In other words, this bit is on and this bit is on and this bit is on, and you combine it and it ends up going somewhere.
Yes, and I will demo that as we get to it in a moment in terms of one aspect of vision. So let me start with the biological plausibility point, and that's going to be in this very quick overview, where the way neurons work is you accumulate charge within a neuron, and then eventually you get enough that the cell fires and the signal propagates down the axon and it influences the input of adjoining cells. And one of the things that's important about this is that we idealize these signals in our mind, but the signals actually look really messy like these in the background. And we are assuming that a spike is a spike, and the fact that these have slightly varying amplitudes, that's noise. And all of this stuff around the bottom, that's noise too. As far as we know, none of that is significant. One of the things that has happened recently is, of course, when a signal enters crosses a synapse to a dendrite, that signal has to be reversed. And so that dendrite has to know when this neuron fires. And so the signal has to go back out to the dendrite in order to reset the synapse. And some people have unfortunately named this backpropagation. And it's a completely different thing than the gradient descent backpropagation. And there is a confusion about that. So when I'm just want to dispel that from the beginning, that because of that, although there is something that is sometimes called backpropagation in neurons, it's a completely different animal than the backpropagation, which is gradient descent that you find in AI.
It would be reset is a better word than signal propagation.
Yes, exactly.
Yeah, that's what I was thinking. It's just like, hang on, you're just resetting the chemical system.
I'm sorry I didn't catch that from whoever was speaking.
Oh, yeah, it was obvious that it's a reset, not
right, exactly.
So who came up with that? Never mind.
Yeah, well, yes, and if I hadn't already had discussions with various people on oh yeah, backpropagation exists in biological neurons, I wouldn't even bring it up. Okay, moving on to task number 2. I guess I can't do it on that screen. I'll do it on this screen. Okay, UKS compatibility. And as I said, I've already encountered a couple of issues for which I had to extend the UKS. This is the Universal Knowledge Store, which is the central data structure with which I'm working. And I think that it pretty well represents how the brain might store and utilize information. And I'll talk about the change that I had to make quite recently. As I say, there's no gradient descent backpropagation. And now we're going to talk about vision. And the reason I'm choosing vision is not because I'm in love with it as a topic, but it's the best understood area of how your brain works. People have actually traced around a lot of things. And I built this diagram that's kind of generally right, that you get some vision input, and then there's a track that detects boundaries, and it's more or less separate from the track that detects colors and textures, and they kind of merge again a little further down the track. But we are sure, we can be confident that there's a boundary and edge detection, and we can be confident that there's these fine segment fragments. And at some point you find corners and arcs, and some point you find edges and areas, and at some point you find, why did it change? It must be on a timer. Shapes and like characters, and you handle occlusion and all of this other stuff. So the key is not what any of these specific steps are, but that some of them are well known and some of them are, and that there is definitely a sequential process, and it's on the order of 10 steps. And that's really important from a timing perspective. And I've put color and texture detection down here as a separate, is kind of a separate track, and it definitely feeds back into areas when areas are found, but it also feeds back into the boundaries, because you can also find boundaries between one texture and another, which is a little more difficult to explain. But don't focus on any of the details of this, just to know that there is this step-by-step process, and it's on the order of 10 steps. And the reason that that's important is that it'll come directly to the timing of things that we'll get to in a few slides. And the next topic on this picture, there we go, is the area of the software that I'm working on right now. Is there is this patch. There are patches in your visual cortex where you can detect specific angles. That is, specific neurons in your visual cortex respond to specific angles. And the total processing time through the whole process of vision is about 100 milliseconds. That is, you can see something and respond to it in about 100 milliseconds, or a tenth of a second. And if there are 10 processing steps, on the order of 10 processing steps, that means you're limited to 10 milliseconds per step. And the problem then becomes that if the maximum spiking time of a neuron is 4 milliseconds, the maximum you can get is 2 or 3 spikes per step. And that says you cannot possibly represent a grayscale or a color or anything like that. You pretty much have a go, no go, that something exists or it doesn't, because there simply isn't time to represent a grayscale or a color, which is why colors and textures and grayscale are kind of running in their own separate path, and they end up merging again later on. But when we talk about finding shapes, and we're going to be talking about that as a first order of business, that you have no alternative but to work with a very small number of spikes. And for a moment, we'll say, well, you just get one, and one means this. One spike might mean this angle, and one spike might mean that angle, but then we'll enlarge on that in a little bit. Are there any questions at this point? Andrew.
Hey, I just it's pretty cool, because I've noticed with the separation and the layers, you know, with the colors and stuff, like if I'm looking around, like I'll see something red, and then I'll like, if I'm looking around like quickly, like I'll see something red, and then I'll see another object, but it's a different color, but I still think it's red. And then I'm like, half a second later, I'm like, oh no, that's blue. So there's like this, I feel like that's the merging, you know, it's like a delayed response.
Right. Another interesting feature of that is that your ability to detect boundaries has a much higher resolution than your ability to detect colors. So you detect the boundary of an object, and then you presume that if you say it's red, that the entire thing is red, even if the boundary of the red and the boundary of the shape don't quite align. You just assume they do.
I think there's another hand up.
Yeah. Mad Max, Richard. You're muted.
Oh, I'm always doing this with Teams. They put the mute
much
in the wrong
better.
place. Yeah, that's better. I'm actually doing something similar. I'm doing it, but on the audio side.
Yes.
So I've taken from what we know about the cochlea and tried to find as much as we can. And go, yeah, we know a lot about what's in the ear, but when it hits the brain, it seems to sort of disappear. Or at least I can't find any more information as a layperson.
Right.
So I've been sort of mentally processing how do I process that type of thing. And I've come to the same conclusion that there's basically so many separate, there's one common source, and there's just so many things that are just plugged into effectively like a bus.
Yes.
And you're saying exactly the same. And then
it's good that you bring that up, because I also presume that the process of recognizing speech or any other sound is likely to come out to be very similar to the steps that we need to recognize an image.
I'm absolutely convinced there is, for like loud noises, etc., things like that. But then we've got the phonemes. We've got part. I'm writing it so that there's a front end which just handles the phonemes. Then that feeds into something further, another system, a layer back. And I got this feeling because of the speed, we've got, even in, I presume you've got the same. Well, you've got the same thing in the vision, haven't you? You've got something right next door to the what I'm calling the bus or the source, and then you've got a processing module behind that, but and so on and so on. Yeah.
Yes, and one of the interesting side effects of that is that if you see the world a tenth of a second delayed, and you likely hear the world a similar distance defined, and then your brain kind of just syncs everything up, and you can't tell until something hits you on the head before you see it coming.
Yeah, I've just got the model I've got is actually it's extending into everything. Is we've just got these sub modules that are just connected to each other.
Exactly.
The scary thing I've got is how do they all communicate with each other? Is there a common bus in the brain? I'm using.
Well, no, there's no serialization. It just it's a combination of firing that produces that maps to something. And if you want to observe how that mapping can be wrong, walk around your apartment, your house at night when the moon is out, and you'll look around and there's things in your area that you recognize during the day and you know what they are. But when you look at them at night, your brain is going to tell you that it's something that it isn't. You're going to witness errors.
No, I get that, but there's so many different systems all over the place. How do they? Anyway, that's not for this. That's a deeper question.
But that's what I said earlier. It's combinational logic. Is that if this and this and this and this are true, then it comes up with the results.
Yeah.
Mm-hmm. And I will show that in a demonstration of the software in a couple of more slides. Signal representation in neurons. You can presume a relatively small number of possibilities. The first is that single spikes have meaning, and that would be my contention most of the time, that an individual spike has a meaning, and the fact that they come in clusters or trains are a necessity of reinforcement and necessary to say, yeah, this is, I want this meaning to be important or this meaning to be less important. And meanings in this instance might be, there is a boundary point at this location, or in terms of hearing, I've heard this specific syllable, or I've heard this transition from one sound to another, which means a syllable. There are pretty much only two ways to represent an analog value in a spike train, and an analog value being something like grayscale or a color or something like that. And that is, you can have spike frequency, and you can worry about the interspike timing. The interspike timing, that is, the spike frequency is, say, over a period of a millisecond, how many spikes occurred. And it's likely that your optic nerve works that way, but it's not entirely known. But the problem of that is that it's too slow. If it takes you 10 spikes to represent 10 different levels, that takes 40 milliseconds, and you know that if you want to represent a gray level in 40 milliseconds, you're never going to get through the vision pipeline fast enough to be useful. And so we know that we can presume that the optic nerve gets itself detected into boundaries and colors and grays levels in different areas, so that you're never having to wait for one or the other. Did that make any sense?
That's fascinating.
It does, but it also makes me think of Brain Simulator 2 and the leakage.
Absolutely.
Because the leakage is really the offset to the timing. In other words, it goes hand in hand.
Yeah, and I actually, let me bring up, since you bring that up, a very short demo of Brain Simulator 2. And it's on a different screen, so I need to share that in this other app here. There. And we're going to zoom in on, and I don't know how many of you have used Brain Simulator 2.
I have.
But these represent individual virtual neurons. And in this area, we are detecting a single spike value. And you can see that this is spiking at a rate of 0.25. And with different, by adjusting the synapse weights or adjusting the leakage rates, you can set up an individual neuron to respond to any frequency you want. But that means that a single neuron is needed to detect every specific level that you want to differentiate. On the other, so if you want to be able to detect 10 gray levels, you got to have 10 neurons running in parallel to do it.
Yeah, but that seems like a reasonable explanation, doesn't it?
Absolutely, but it has some implications for things that might be happening. That why it is that you can only detect a limited number of different gray levels, yet you can detect the boundary between two gray levels with a great deal of precision, because there's a different mechanism that will detect the difference in two firing frequencies, as opposed to detecting an absolute firing frequency. And so you can end up with gradients where you don't detect where you don't ever detect a boundary in the gradient, or in some areas you can set it up so you can, and you end up with interesting optical illusions because you can detect differences in firing rates or boundaries between two firing rates with much higher precision than you can detect the absolute firing rate. Did that make any sense?
Yeah, well, one is like classification, and then the other is more of a continuous analysis.
Right. And so
I was just about to say, you're detecting the difference between a frequency change and the absolute values. It's just the same. I mean, one is easier to take to the detection of the frequency difference. That's where the boundary is.
Yes, you can detect the frequency difference and detect the boundary, even if your ability to detect the two sides of that boundary appear to be exactly the same color.
Because they won't have the same wavelength, so the intertiming timing is off.
Yeah, but isn't the result ordinal? Isn't that all you're detecting, is that one is greater than another or less than
Yeah,
another?
but that's all you need. For a
Right.
boundary, that's all you need.
Exactly. No, a lot of things in decision making are ordinal. In other words, if you're going to a restaurant looking at a menu, you're really only ever going to compare a couple of items at a time. And then you're going to say, okay, well, I prefer this over this. And then I prefer this over this. You can't consider 40 items all at the same time.
I mean, I know in the ear, we do get frequent firings and number of peaks based on the volume. But I have always presumed that the very first fire on a particular frequency band is actually the key thing to trigger things. And then the subsequent firings are therefore slower processes to pick up, if that makes sense.
Yeah, and this touches on an interesting separate problem, and that is the maximum firing rate is much lower than the propagation delay. A signal can get from an input to an output to an output synapse in maybe 2 milliseconds, and yet the neuron takes 4 milliseconds for it to reset itself so it can fire again. So that has some limitations. Okay, let me move on to the next question, which is, in your eyesight, and I'd like you to give yourself a demo, you have much more limited resolution than your mind thinks you do. And we measure the highest resolution at the highest resolution area of your eye as about 1 arc minute, or 60 pixels per degree. And excuse me, let me offer you a demo that the size of your fovea is about 2 degrees. So, and that's about your thumb width arc at your arm's length. So if you back up away from your screen to a point where the text becomes just on the edge of hard to read, and you hold up your thumb to block out some set of characters, you find out that your thumb will block out about 10 characters. And so since we can do a little arithmetic and see that the actual high resolution area of your vision is about 120 by 120 pixels, and that means that you've got 12 pixels per character. And then you scratch your head a little bit and you say, that's pretty crummy resolution. It sure seems better than that to me. And I think that we're going to find that the 2 or 3 spikes that you might possibly have in a level, rather than representing a gray level or some other transient, represent offsets from the center. So if I have 120 by 120 pixels of absolute resolution, if I say I get 2 spikes, that means that the boundary is right on that pixel. But if I get 1 spike, that means it's half off towards the next pixel over. And so you can get that to expand your resolution without slowing the system down. Did I say that in a way that made sense?
Yeah, but isn't it, you're building up this memory of what you think? For instance, don't you have a blind spot in each eye where the optic nerve comes in? And
that is true, but that's a separate question. The real deal is that the high resolution area of your eye is so small that as your eye darts around, it builds up this map that you believe is high resolution all over, but is only high resolution at the area you're currently looking at. And you remember all this other stuff. So as you're reading text, as you're holding your thumb up against versus your screen, you'll notice that you really can't read the text on either side. Or if you do read the text on either side, it's because your eye moved.
Sure.
And,
But
and
isn't it just building up? It's building up that model in your head in
yes,
pieces and
yes,
retaining where you looked recently.
yes. But I think we'll find that you build up the model in terms of things that you've recognized, not in terms of individual pixels. So you'll remember that you got the first half of this word and then you remember the second half of that word. You don't remember the individual pixels that were involved in that reading process. And so this is a huge data compression mechanism so that you never have to recognize the 1,000 by 1,200 image that your screen shows you, because your eye darts around and you don't even notice it.
Right, but that's all downstream, so that indicates there's a hierarchy going on.
Yes, absolutely. So the visual cortex corresponds, the V1, the first area of visual cortex, corresponds to absolute areas within the brain, within the retina. So the high resolution of the retina is represented by a specific high resolution area of the visual cortex. And this area that the mental model that you have of your surroundings where you're filling things in is at lower resolution because it is in terms of the objects that you saw there, which is a much smaller suite of things that could be happening. Okay, let me show this in a demo. Excuse me. I'm going to run two demos actually, and we're going to do that on this screen here. There. And we're compiling. We're still compiling what's broken.
Oh, those magical words.
I think we finally slowed this little machine down with between the video of the meeting and the compiling and all this other stuff. I'm going to show first software, which is a previous version that I built. And I've written this vision system a couple of times, and I'm finally homing in on what I think must actually be going on. And here we have a representation of a character, and you can see that this is right out of an MNIST kind of a presentation, except this is a quality character. But when I turn on the boundaries that are detected, you can see that the boundaries don't necessarily fall in the centers of the pixels. And what that means is that your eye is capable of recognizing boundary zones at a much higher resolution than the input image. The input image is like the original JPEG that we're seeing the 4, where it's got grayscale around the edges. Well, your eye sees that grayscale around the edges as the boundary that it determines is the best location in between the white and the dark, or based on the gray level. And so the boundary pixels don't fall on the centers of the pixels. And this is a little different from the way AI vision typically works, because AI vision typically starts off with a few levels of convolution, and the convolution requires that the pixels end up being right on the detected boundaries are on pixel locations rather than wherever they want to be. And I think that this is going to be an interesting idea. And this, by the way, is an algorithm that is, well, it simply shoots rays horizontal and vertical, and it finds the best location for a boundary. And it works pretty well. And boundary detection actually occurs in your retina. So by the time the signal gets to your brain, you already know where the boundaries are. And so this is the current software that I'm working on. And on the left, we have the representation of the UKS content. And on the right, we have a representation of your visual field. And at this point, I'm just going to be showing a single patch cell. And if we initialize, and I'll talk about that in a moment, remind me if I forget. What the initialize has done is and here we are in the UKS, it's created a set of boundary points that are just labels for convenience. And then it has patches. And I can click here, line, and it'll create a line and show me the patch. This is the patch. And the colors are representative of the weights of the connections. And so I can refresh this, that is, present the same image again, and you can see the weights to the connections subtly changing, and you can see these edges have actually gone negative, which I want to talk about in a moment. But if I say show will show me the other entries, others are blank. But if I say run 100 lines in, and it'll take a second or two with the video running particularly. There we go. And the patterns that it's detected. So it's learned these patterns of the various lines. And this is akin to the slide that I showed with the ability to detect various angles at various positions. And so this, after boundary detection, this is going to be the first layer of defense, where in your eye, the idea that the patches are square is pretty crazy, but it's more likely that they're rounded or have rounded corners or something like that. But the way this works is I can now show an individual line. And this individual line is not through the center of the patch, so it's not detected as anything. It's going to be incumbent on the next patch over to find this one. And this is just a fragment that doesn't find that. It finds this fragment because it goes to the end. And the next layer of processing is going to take the outputs of these cells and make them into an array of detected line fragments. And from line fragments, we should be able to find corners. And from corners, we can begin to build up areas and shapes. So this is the first stage. And I have only built it on one patch at the moment. The code all exists to make multiple patches, but I haven't explored it. But my expectation is that because I have boundary detection, that I can feed this random images, and you can see this, I only gave it 100 presentations of various lines, and it did a pretty good job of detecting every available line that it could possibly get in this visual patch. So I think I can give it random images and get similar results, because I'm looking at a patch that's quite small relative to the size of the image. And if you're only looking at 5 pixels, if you have a couple of boundary points, they probably represent a line or perhaps an arc. And one of the things about having an arc is that means that the end pixels have to have lower weights, so that the further you get from the center, the more tolerant you are of errors. Um, is that making any sense?
Well, that's interesting because if you look at the MNIST and how badly shaped some of those characters are,
Yes.
you try to think of rules that would describe it, and it's very difficult to sort of, and that was not successful. The deep learning was eventually what came in and produced a result. But
Right.
what you're saying here is that, well, what matters, it's not the rule, it's you have to consider the distance from sort of some center point.
Yes and no. The key is that you'll have an array of 120 by 120 of these cells. And by the way, these cells are all represented on this side. So if I clear out this pattern, I can say, show me this patch. And that's what that patch is. And so this is all UKS data displayed in the right-hand side. I didn't clear it that time, but so I'm looking at essentially the 8 cells that would be related to an individual pixel and its surroundings, where it's looking for a boundary at that individual pixel. And in order to have a boundary, you have to have at least one boundary point, probably 3. That if you don't have 3 boundary points in a line, you probably don't have a boundary. You probably have a speckle of a texture that
But how would this apply to really poorly formed digits in MNIST data set?
Okay, even in the poorly formed digits, you find the boundaries and you detect the angles at the boundary points. And at the next level, we'll turn these into corners or arcs. And in any real world, almost everything is a rounded corner, because square corners are something that we have in technology, but they don't really exist in nature. So your eye is attuned to finding rounded corners, where you will find this cell and the adjacent cell has a vertical line, and you'll say, aha, there must be a corner there, because you've got a change in slope, must be a corner. Did that answer your question?
Well, I'm just trying to think of, because I mean, if I actually looked at, went through the digits in the MNIST, and some of them are really bad. I mean, they're very poorly written, and you can still, some of them are not. I couldn't figure out what they were. Even though they said, well, this is a 7 or something, it didn't. Yes. I didn't think it was a 7. But there are, there's different angles. And then also, somebody might write something with a very sharp pencil, and somebody might write it with a marker. And the marker has it's a much bigger swipe along the paper, and so that also makes it harder. I mean, in terms of thinking of rules, but the brain seems to do it okay. And I'm wondering how, if you sort of zero in on these little subsets, how do you join these subsets to conform to one rule set?
Yes, one of the key. The first thing you have to do is to match up the boundaries to being opposite sides of a stroke. So you have to think in terms of strokes, and then the strokes have corners, and given the corners and arcs, you can look up the UKS. I have done this, and it works pretty well. Given a set of corners and strokes, the system can return the most likely character.
Okay.
One of the downsides of MNIST is that in advance that it's a digit. a lot of the stuff that you see in MNIST, if you didn't know it was a digit, you'd have no way of knowing what it was. Things like ones and slashes and minuses, they're entirely interchangeable in the MNIST database.
Right, but what you just said is very interesting, because you're saying, you talk about arcs and that you're really talking about relationships in an abstract sense as opposed to a specific sense.
You have to. Otherwise, if you move your eye, you can't see it anymore. Or you have to have multiple copies that for every position in your fovea. And that is implausible to me. My suspicion is that you have a dozen different twos of different shapes, and your eye simply matches, your visual system matches corners and arcs to fit the set of the two, rather than having to move things around in order to find them.
Right, now that makes sense. That's very interesting.
Yeah, well, it's the only way I can see that it can work.
But the problem is always, if you're trying to come up with specific rules, it's impossible. Yes. It's impossible. And what you're doing here is you're saying, okay, well, we have these sort of general building blocks, and they sort of fall into discrete buckets because of their relativeness.
Mm-hmm. Another thing that's important here is that the further you get from the center, the less your weight is. And so the more tolerant the system is of error, the further you get from the center. And that's an important key, and that'll be coming up in a moment as something that is really important when I get to the next screen here.
I was just about to say, wouldn't the fact of some patches being erroring out because of artifacts in the visual system or something like that still means that at least you've got some fairly strong probability going through the different layers back to say that that's too. So yes, you could just totally ignore that patch because that's just random for whatever reason.
Yeah, and this is of course complicated by the fact that if you have textures, you have the polka dot dress, you have no trouble finding the boundary of the polka dot dress. And so that has some implication as to how you process textures and patterns as opposed to other things, as opposed to hard-edged characters.
How would this relate to occlusion? In other words, if you're picking oranges and it's occluded by some leaves,
okay,
how would this fall into that?
About 5 steps further along the pipeline, when I will have an arc and a corner and an arc and a corner, I will say, I will match this against my library of known images, and I will get back a match on 2 of the corners, but not the arcs because they're occluded. And it's going to give me the most likely match.
Okay.
And there has to be some mechanism by which you process the thing that you perceive as closer first. So if you're occluding the image by your hand, that you process the hand first. But I want to go back to just a couple of other things, and that will be on this screen again. The conclusions that I've drawn, and there are a couple of really interesting things that I didn't know that happened. The first is, in order to make this work, I had to have a nearness gradient. That is, the closer you were to the center of the patch, the more important your pixel value was. And in order to make that work, you have to have a maximum relationship weight for every connection you have. And the UKS didn't support that before, and I had to add it. And the reason is that given enough learning, all of the pixels will approach 1, all of the matching pixels. And that gives equal weight to the ones that are further away, where you're more likely to get errors. So you have to be able to set maximum values. And I thought about how that might work in a physical neuron system. And this will be something for the neuroscientists to actually go and explore and look at. So we have a testable hypothesis here, and that is you will assume that the physical maximum of an individual pixel individual synapse is on the order of 0.1. And if you want to have a connection that has a maximum of 0.5, you got to have 5 synapses in parallel. And so we should be able to look in the visual cortex and say, in the centers of these angle detector neurons, that they have a higher concentration of connections at the center of the patch that they represent. And I think that's going to be useful, that the relationship maximum is proportional to the number of parallel synapses, because we don't grow synapses very quickly. So let's imagine that the system is fixed. Another problem I ran into is with inhibitory synapses, that it's easy to see that you got to have negative weights for the synapses that you don't want to fire. And the problem is that all of the published learning mechanisms require that both sides of the synapse have fired, and you use the relative timing to decide if you're increasing or decreasing the weight. But what we need to have is a negative synapse that increases its weight if the target fires, but the source did not. And we should be able to poke around it. That is, the neuroscientists should be able to poke around at synapses and find that this is the case, because there's no other practical way to make it work. They do know that you can set an inhibitory synapse by firing the target shortly before you fire the source. But that means you still got to fire all those black synapses, and that doesn't make any sense to me. That we've got to have a system where firing the white synapses is sufficient to get you to recognize the picture. The current patch algorithm, I have written this in such a way that that you can build an array of these patches, and it'll happen by magic with a given stride and all of this. But I just didn't have time to try it out. But my suspicion is that given any set of images with any kind of boundaries in them, using my boundary detector algorithm that is kind of homebrew from elsewhere, if I get a set of boundaries that I can build, that I can learn how to fire these angled patches autonomously, that I don't have to do anything. And so my suspicion is that your visual system doesn't have to have any of that information, that after a couple of days of vision, your visual cortex will build it automatically, which would be nice to know. I wanted to talk just a minute about the code behind this. And I have code over in this window over here. And let's, uh-oh. Is there, I hope we're still there. Here we do the array initialization. And all this does is it creates a two-dimensional array of boundary points. And then it creates the patches with some set of stride and some set of weights having to do with that. And this small amount of code where it sets the weight of the individual connections, that's what has to be in the DNA. That if you set that up in the DNA, then you can repeat that process ad infinitum, and you'll end up with the beginning of a visual cortex. And my suspicion is that we'll find a similar rule as we add layers and steps to the processing of the visual cortex. But I have no evidence about that yet, but I hope to hear in a month or two. And second, I wanted to show you the learning process. And here, where are you? Search and learn. And what's important here is that it calls search for closest match. And search for closest match is the same algorithm that is called if you want to know what has 4 legs and a tail and get Fido. It's exactly the same algorithm. So I'm using the same search process in the graph structure to find boundaries and small segments that I would be to be following, finding 4 legs and a tail. So the logic is the same, and I think this is going to be really interesting to find out that the search for closest match is exactly the same. And then when you find the match, I've got this adjust weights thing, and the adjust weights thing comes down to these 3 lines of code that if you have a relationship in your patch that goes, and it comes from an individual input pattern, and you ask here, is the target and the stored pattern, does the stored pattern in the patch match the pattern in the target, the search target? And if it, to the extent it matches, then R found will be true for that individual pixel. And if R found is true, you increase the weight. And if the R found is false, you decrease the weight. And this is so dirt simple. If we can apply it to multiple levels, it entirely replaces backpropagation. We'll see how that works.
Well, it looks like you have a discrete algorithm as opposed to a continuous one.
What do you mean by that?
Continuous is in backprop. You look at the derivatives, and you use
that. You're absolutely.
Whereas in discrete, you're looking for events as opposed to measuring the slope of something.
Absolutely, and we know in neurons that learning happens with proximity of firing. Here's where I mentioned that I have to be able to set a negative neuron if the input doesn't fire. Well, okay, we'll just have to search around, have to fire up the neuroscientists and hope that they can find that. And then, but it increases to the max weight. And it turns out if you don't have a max weight, this thing, the whole idea of this collapses. And so I had to go back to the UKS and add a max weight parameter to the relationship because nothing was going to work without it. And I thought that was kind of interesting. When you try to do something, you learn why it doesn't work, and then you fix it. And this is more my evolutionary coding, which except it's manual instead of automated.
Well, it's similar to saturating a neuron, right?
Exactly. And the key is, though, you've got to have a maximum somewhere.
Right, but neurons have maxes. They either fire or they don't.
No, the synapse weight has to have the maximum.
Oh, okay.
And I have presumed that the synapse weights all have exactly the same maximum, and yet your DNA can construct your visual cortex so that it has different different numbers of connections. And so you have connections in parallel that emulate a higher maximum.
Sure, but that I think that it really is an event-driven type of learning.
Yeah. And we do know that in neurons, learning only happens when the neurons fire. If no neurons fire, you never learn anything. Of course, you never do anything, but you certainly never learned anything. And so I've kind of got this code that kind of marries the UKS and the idea of how individual neurons might be working in the visual cortex. And it's my speculation that that you originally build out cells in your brain in such a way that they represent, that they have all of the capabilities of UKS cells. So they can learn and have backwards connections and all this other fancy stuff. And in areas like the visual cortex that are quite specialized, once it builds its structure up, then all of the unused stuff that is overhead for the UKS kind of atrophies away because it's never used. And the neurons that are never used get repurposed and reconnected into something else. And so you end up. So yeah.
Just
want
to
Mad Max.
Yeah. Just want to throw something in here. The eye has evolved, as I said on Discord. I'm a paleontologist. Well, I was a paleontologist that trained. The eye has evolved. We know this separately 9 times throughout the whole of evolution. Yeah. And you can look at the eye of the cephalopods. It's totally different. Look at the eyes of insects, etc., etc. But they all have the same neural. They're all basically the same neuron, aren't they?
Yes.
But also, the other thing is to take into account is a lot of animals just work in grey or limited colours. Some they'll do we all seem to do intensity. Every animal seems to do intensity. And then it's colour added on, isn't it? But then others also have polarization.
Mm-hmm.
I'm just wondering if those things are just they're just sort of like parallel channels, as like you're talking about.
Yeah, I think that's an excellent question, and my guess is that generally this is the only way it could possibly work. Yeah. And that now you can make some changes in the specifics so that you are sensitive to color or not sensitive to color, or sensitive to this and not sensitive to that. It's been, but my expectation is that we'll find as we move along that there's a way to build out most of a vision system this way. And it's not important to me that I have a vision system, but it's important to learning what these general rules are. And I'm learning that that the structures that you need between one neuron layer and the next, or one set of graph nodes and the next, are predictable and fairly simple, and that the learning process is also fairly simple, and that to having made this work this far with this really simple stuff has been pretty gratifying. Okay. Are there any final questions, observations, comments? Okay, well, I want to thank you all for coming. I noticed Fry's joined. I'll catch him up in a moment. But I appreciate everybody here. In a couple of days, I'll have this massaged into a video. I think that it's going to be, well, for me, this is fascinating stuff. So I hope that it's at least interesting to you guys as well. I appreciate your coming in. And if you can invite your friends, that's great. I appreciate it. And if any of you want to participate in writing some code, I'm happy to talk to you about it. figure out some places that we can coordinate. This is a little different than most coding processes because I don't know what the answer is. I don't, and so I write some code and I say that doesn't work, and we'll write some new code. So, and I don't know how you distribute a project where it's being written that way, but I'll give it a shot if you're interested in writing some code.
That just means there's no wrong answers.
Yeah, it's all hacked together.
Exactly.
Hey,
okay, well,
it's the rule of three. The first one you do is your prototype because you have no idea. The second one you do is there, but there's bugs, and the third one's perfect.
Yeah. Well, on that happy note, I appreciate all of your coming and time and your comments and questions, and I look forward to writing some more code and putting together another group like this in a couple of weeks, and we'll see how that goes. But the key is that I want to have something to show before I schedule a meeting, because we don't need to go back and rehash all of this stuff again. I thank you all for coming. I thank you so much.